Hello, everyone, and welcome to today's live broadcast, Integrated Interrogation of Oncogenic Signaling Pathways, Immunology, and Heterogeneity in Cancer, presented by Michael Davis and Joseph Beecham. Dr. Michael Davis is the Deputy Chairman and Associate Professor in the Department of Melanoma Medical Oncology at the University of Texas MD Anderson Cancer Center. Dr. Joseph Beecham is Senior Vice President of Research and Development at NanoString Technologies. I am Marjorie Torres of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by NanoString Technologies. For more information about our sponsor, please visit www.nanostring.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask the Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom or center of your screen, or use the Ask the Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Accreditation button located in the promotional board at the bottom or center of your screen and follow the process to obtain your credits. Please join me in welcoming Dr. Michael Davis and Dr. Joseph Beecham. I will now turn the presentation over to them.
Thank you very much. This is Joe Beecham. I lead R&D for NanoString, and I'll be introducing you to a brand new platform that we've recently developed called the Digital Spatial Profiler. I'll give you some background on how we developed the microscope, and then I'll hand it over to Mike, and he's going to describe to you exciting new things that are actually happening on this microscope in the area of cancer immuno-oncology. So just as a brief introduction to NanoString Technologies, we develop methodologies that allow you to perform assays on proteins, single nucleotide variants, gene expression, gene fusions, copy numbers, single-cell gene expression, as well as microRNAs. Our technology is highlighted by being able to analyze up to 800 targets at a time. So it's a multiplexed assay that measures 800 different things. And a little snapshot of the view of the current instruments. These are not the brand new microscopes we'll be talking about. Our current instruments that run these assays are shown there in the upper left portion of the screen. That one there you're looking at is actually called the MAX. Let me describe to you a little bit about the background on NanoString and how we actually make measurements. We make measurements something that's called single molecule optical barcode counting. And it sounds like a mouthful, but it's relatively straightforward to understand. What you can see on the screen is a whole series of things that look like balls. There's actually 6 of them there, and each ball can be one of 4 colors. And the key to a nanostring, and that's the nanostring in our name, is that the pattern of colors represents the actual analyte of interest that you're going to be measuring. So it's going to be an automated microscope. It reads that barcode, and it determines the digital counts associated with either mRNAs or proteins. So by just changing the color positions of those balls on the end of that particular, you can see that small piece of 50 base pair nucleotide overhang, you can index into up to 800 different things. So then when we make our measurements, our technology is amplification free. It is single molecule direct digital counting of the analytes of interest. So the nature of our data, it's all completely digital. And it's highlighted by not using enzymes or amplification. So we want to measure something. We actually generate hybridization complexes that actually will take these particular reagents that are designed to measure up to one of 800 different targets. And when those reagents that we make here combine with the nucleic acid, and you can see that there in the middle of the screen, when the nucleic acid comes in, it hybridizes with both sort of the sticky end of the optical barcode, which you can see to the right, and the little white ball on the left-hand side of the screen is actually a biotin molecule. So we mix these samples together. The measured nucleic acid that came out of the cell of interest or the tissue of interest forms a hybridization complex. It combines with the single molecule optical barcode that allows us to see a single molecule, and then we digitally count that. And that's the nature, the intrinsic nature of our detection chemistry. Now, these barcodes that we've developed, and this is an important work that we actually did a long collaboration with MD Anderson, where Mike is on this webinar also came from, is that we actually advanced this barcode technology so we not only measure RNA expression, we can also measure single nucleotide variants in DNA, and we also can measure protein single molecule counting also. And you can see for the little trick we did to measure proteins digitally, since they obviously do not have nucleic acids, is that we actually take antibodies, label them with a small 60-mer single-stranded piece of DNA, and then we count the single antibody through the hybridization complex on the tail of the antibody. So that's how we do the protein-based detection. Now, one of the areas that motivated us to develop this high-plex microscope is it's wonderful to measure 800 analytes in solution and determine the properties of the tissue under investigation. But one of the key aspects of tissue is actually the tissue structure itself. So instead of doing sort of grind and bind assays where you just scrape off the tissue slice and you measure 800 analytes, which is a very, very useful thing, and it's worked very well for us. There's certain problems in biology that you actually need to maintain the tissue structure. Now, the trouble when you want to maintain tissue structure and actually see a tissue, you go into a microscope. Now, the problem is when you go into a microscope, you're typically, instead of measuring these beautiful 800 different analytes at a time, I'm sort of stuck with only measuring roughly 1 to 4 analytes at a time. So that was not very satisfactory to either me or Mike. He has all sorts of questions, and he needs to look at more than 4 pieces of information at a time. So we actually invented a microscope that was designed intrinsically to be a high-plex microscope with spatially resolved capability. And this is the design strategy we used. We built a microscope that's totally different than anything else that is out there. Everyone else is trying to put all the assays for their components into the tissue and observe it in the tissue and the microscope at the same time. That's what limits these assays to roughly 4 different colors that you can observe at any given time. We took a totally different approach. So what we did is that instead of our imaging reagents, or instead of our reagents that are actually our affinity reagents that are going to go in there and measure mRNAs and proteins in a tissue spatially resolved, instead of putting the colors on our reagents themselves, which would limit us to like 4, we actually did it a different way. All of the measurement analytes that we have in this new microscope that we built are actually on either antibodies or on the FISH probes, and you can see that in the lower left-hand side of this slide. And they're on those affinity reagents with a special kind of linker called a photocleavable linker. And what this allows us to do is that we can go ahead and take a single slide and pour as many as up to 800 antibodies, or probe as many as 800 different mRNAs, and we can put those affinity reagents on the tissue slice itself. And then when we go to read them out, instead of actually trying to read the detection with colors on the inside, we move the microscope objective over the region that we want to examine inside the tissue slice. That's called a region of interest. When we find that region of interest, we do a brief burst of 365 nanometer light, just on for the order of about 200 milliseconds. And what that does is it releases the photocleaved oligonucleotide tags from either the backs of the antibodies or from the mRNA probes that are in there. Those photocleaved, and you can see that right in the middle screen, you can see the photocleaved oligos escaping from the tissue surface. And then as they escape from the tissue surface, the microscope is designed to literally drink them with a microcapillary right off the tissue surface onto a microtiter plate that is on the automated microscope. What this allows you to do is you're making what I call a digital replica of either the antibody distribution of any region in space or a digital replica of the mRNA in a region of space. And that's highlighted by the right side of the panel. You can see those small circles and things. Those are regions of interest on a particular tumor site that we're investigating. And then we, with a microscope objective, would have gone underneath one of those little regions of interest, shine light. The barcodes would come off the surface, We digitally count them one at a time, and you end up with a heat map of digital counts linked to each one of these individual regions of interest. And in this particular manner, the microscope can digitally count single molecules at a time with up to 800 different analyte classes, all in a small region of interest of the tissue surface. And the regions of interest can get very small, all the way down to single cell. So when you actually perform such an experiment, you can see here, in this particular case, we have an excisional biopsy. So this is a tumor biopsy here. And you got highlighted in colors actually two items. What you see in purple are actually the tumor cells that are lit up. And then the area that you see in green is actually CD45. And this is like the immune infiltrate that's coming in and starting to penetrate this particular tumor of interest. Now what you can do is, in a typical microscope, you would just be limited to these kinds of colors. But now we want to go in, and you can see here, let's just take a look at that region number 10 there with that little small white circle. When I'm in the tumor region, I shine a brief burst of light and then count the antibodies that are located in that tumor-specific area, and then I count them. So that you can see that when I'm in the tumor itself, markers that are high in tumor but low in immune cells show up. So you can see the large spike in pancytokeratin. You see that large purple ball to the right, and that number is very high when I'm in the tumor environment, so when I'm in the purple area and shine my light. But if I move the microscope objective over to region of interest 7, which is now in the immune infiltrate region, and perform the same experiment, and I look at what antibodies are in that region, you can see there's almost no pancytokeratin over there because most of the tumor is not actually in that region of interest. But what you get is an increase in all the key immune cell markers that are localized in that particular area of the immune infiltrate. So this is just sort of a pictorial display of how these kinds of experiments were done. And Mike will explain to you exactly the types of very interesting immuno-oncology questions you can ask and probe of tissue slices in this manner. So this is the highlight, again, the overall flow of the microscope. There's the tumor section on your right. You bathe it with a whole series of antibodies or mRNA fish probes. You shine light and you count. These are the detailed operations that I showed you schematically and I described just previously. There's your microscope slide in the center. You shine the light up. That will be step number 2. The barcodes come off the backs of the antibodies. They're drunk off the surface of the tissue slice with the microcapillary. That'd be step 4. Those barcodes are then placed in a microtiter plate that sits right in the center of the instrument there you see to your left, the digital spatial profiler. And then those photocleaved oligonucleotides are dropped in, say, position A1 of a microtiter plate. This entire process there from step 3, 4, 5, and 6 takes about 30 seconds. So then you actually just select, you go back up to step number 2, you select a new region of the tumor that you want to probe, and you repeat. So you fill up a microtiter plate with a digital replica of the protein distribution in space on the tissue surface. Now, one of the key aspects of this microscope that's rather interesting to describe, it is a read-write microscope in the sense that I am writing with light, and wherever I write with light, I'm actually reading all the digital barcodes that come off the surface at that particular location. So you can do some very, very unique types of experiments with this configuration. And the writing element is a totally generic writing element. This little thing that you see labeled number 10 is something called a digital micromirror device. And a digital micromirror device can shine light in any specific location in the field of view of the cell. You can see it's a colon cancer sample there to your right. And that light that I want to read can be focused all the way down to a single cell or whole groups of cells in a very, very flexible manner. Now I'm going to highlight just one aspect of the flexible manner of the microscope, and I call it reading and writing in the language of cancer. And this is a unique ability of this particular microscope, the digital spatial profiler, and I'm going to highlight it here. This is a colorectal cancer sample here, and I'm staining it with a dye or an antibody that actually is specific for the tumor cells themselves. That's pancytokeratin, and I'm going to image that with the microscope in the color yellow. Now, a very important answer and question you want to ask out of tumors is that I want to know what the pure signal is associated with only the tumor cells. And what this allows you to do, you can see in that particular colorectal cancer cells, actually about half of the cells you see in that section, the purple ones, are actually immune cells. So if I just took an average signal, I would end up with 50% tumor cell signal, 50% the tumor the host immune cell signal, and they're totally convoluted with each other, and I don't actually know what's the origin of those particular signals. Now, when I run the microscope, though, I can I record the pan-CK image and then selectively write with 365 nanometers lights only where the pan-CK positive cells are, and that is the mask that you see generated onto the right. That mask goes directly and drives the digital micromirror device, and at a single cell resolution, it will actually shine light only on the tumor cells. And then when I drink the barcodes after exposing to light of that particular, looks like it's a complex mask there that you see on your right, the black and white. But when I shine light like that, I know that every barcode that I'm going to be reading is actually from the tumor-specific cells without a contamination from the immune cell component. Now, it's a very useful thing. So it's like we talk about that. Any tumor can be made 100% cellularity with light, and then I get a pure tumor signal. Now, just as valuable as I can take that inverse, if I take that mass with a tumor cell and insert it, and now you see that on the right-hand side, and write on that same exact tissue slice, now what I'll be doing is collecting only the antibodies that are in the immune compartment of that particular tumor slice, and then I end up with a signal that is more 100% related to the immune cells. This is a very unique feature of the microscope, so that in two 30-second operations, I end up with one well with a digital replica of the tumor-only profile, and then the microtiter plate well right next to it could be 100% of the tumor microenvironment. So I'm going to summarize the overall intrinsic features of the microscope here, and then I'll hand it over to Mike, and he'll show you what you can actually do with this on real clinical samples. So one of the advantages of the digital spatial profiler, the first is that it's highly multiplex. So instead of having one slice of tissue and only getting 4 answers, now I have one slice of tissue, and you can get up to 800 answers at a time. The other aspect is the quantitization. So normally in a microscope, you actually measure something and it's like, well, how bright green is it? And that's a very hard thing to quantitate. And oftentimes when people look at those kinds of images, they'll literally do things like that's a 3+ signal of how green that particular area is. Not a very quantitative technology. But since we are actually counting, single molecule counting the digital replicas from those areas, it's the ultimate quantization. This is single molecule counting with up to 6 logs of dynamic range. So you don't have to answer, well, it's kind of bright green plus plus plus. You literally say how intense is that signal? Well, that signal is 23,412 because I counted 23,412 oligonucleotide tags from that little region of the tissue slice. So it's absolutely quantitative. It's single cell, so you can actually focus the digital micromirror device and actually probe single cells. Obviously, some markers would be very difficult to detect at single cells, but not all markers. So there's many proteins that you can observe at the single cell level. One aspect that's also very interesting, I didn't describe it all during my part of the talk, is that's totally non-destructive. The sample has never been touched by anything other than light. So at the end of the entire multiplexed assays we're doing, you can actually take that tissue slice out. You strip off the original antibodies that you are probing it with. You store it in a wet medium, and it's literally totally non-destructive. You look at the tissue, you can never actually tell it's been measured. You could hand it to anyone else, and they can do any downstream procedures they wish. But often of interest is that you may make a discovery on that particular slide. You're going to come back and actually re-examine it with a whole new series of analytes, a whole different set of antibodies, or a different set of mRNAs. You can do that because it's totally non-destructive. It's very unique in that manner. And then lastly, but also important, is that the throughput of the microscope is in the right kind of speed range. We basically can do 10 to 20 full slides a day on this microscope in a standard 8-hour, 10-hour work day. And this is the kind of throughput you need if you're going to interact with someone like Mike at an MD Anderson where they're doing clinical trial programs, and these are hundreds of patients oftentimes that are in these clinical trials. You have to be able to look at 100 patients that are patients, maybe 100 controls, and you have to be able to accomplish that kind of study in a week or something. You can't take months to examine those many tumors. So we decided it importantly to have that kind of throughput for a normal sort of pathology application. Now I'm going to hand this back over to Mike, and he's going to describe to you exactly the very exciting work that he's doing in immuno-oncology using the digital spatial profiler. So over to you, Mike, please.
Thank you very much, Joe, for that introduction. And I've titled my presentation today, Integrated Interrogation of Oncogenic Signaling Pathways, Immunology, and Heterogeneity in Cancer. This slide shows my disclosures. I receive research support as the PI for my institution from several companies, and I've also served on the advisory board for several companies. Melanoma is the most aggressive form of skin cancer, and for decades we really had no effective therapies for this disease and very little understanding of its pathogenesis. And what we've really seen in the last 10 years are really multiple breakthroughs in the understanding of the biology of this disease. And interestingly, these breakthroughs have really happened parallel in two different areas. And so first, we now have a much better understanding of the molecular drivers and pathogenesis of melanoma. This figure shows the melanoma TCGA, which demonstrated by whole exome sequencing that on average melanoma has a higher mutation burden than any other common solid tumor. And in actual fact, in the majority of these patients, we can actually identify clear oncogenic drivers that are present in each patient's tumor. On the right is illustrated the fact that we've also seen multiple breakthroughs in our understanding of the key regulators of the anti-tumor immune response in this same period of time. Now again, as a researcher, we love to see breakthroughs in understanding, but what's been so rewarding in this field is how this information has been translated into new therapies. And so really up until 2011, the standard of care for a patient with metastatic melanoma was enrollment onto a clinical trial because of the futility of the existing FDA-approved therapies, dacarbazine chemotherapy, which has a response rate of only 5 to 10%, high-dose IL-2, a therapy that was able to cure patients about 5% of the cases, but also a treatment-related mortality rate of 1 to 2%. And then between 2011 and 2016, we saw the approval of 10 new medications for patients with stage 4 melanoma, all of which have had significant impact on overall survival. And interestingly, not a single one of these medications is a chemotherapy. Instead, they've been divided between targeted therapies for patients who have a BRAF V600 mutation, which is the most common oncogenic mutation found in this disease, present in about 50% of patients. And we now, with our current targeted therapy combinations of BRAF and MEK inhibitors in patients with BRAF mutations, see response rates of almost 80% and disease control rates of greater than 90%. And patients on average respond to these therapies for a year. In parallel, we've had the development of multiple new immune therapies, starting with ipilimumab in 2011, the approval of single-agent PD-1 antibodies, pembrolizumab and nivolumab in 2014, and then most recently, the ipilimumab-nivolumab combination. These immunotherapies over time have been observed to be more and more effective, with response rates with ipilimumab of 10% to 40% with PD-1 antibodies to almost 60% with the combinations. And impressively, the median duration of the responses with these immune therapies tends to be quite long, actually difficult to estimate at this point in time as our data is still relatively new. And so our field has really been completely revolutionized in the treatments that are available and the outlook for patients. And actually, as researchers, what's really emerged as the dominant question now is how do we actually personalize our therapies? How do we match each patient with the right treatment? And in particular, how can we predict, understand, and overcome resistance? And so with these questions, there's a tremendous need for new biomarkers to help us guide and personalize therapy for patients. And as we've, again, worked on this area, we've identified a number of key challenges that need to be addressed. First, the concurrent evaluation of molecular and immune markers. Again, we've had multiple breakthroughs in the understanding of molecular biology and in the immunology of this disease. And interestingly, what we're learning is that these two processes are not separate, but actually closely regulate each other, supporting the rationale for their concurrent evaluation, as I'll describe. Secondly, we're starting to appreciate much better the heterogeneity that we see in molecular and immune features, and this includes between patients, between different tumors in individual patients, and even at the individual tumor level with intratumoral heterogeneity. And finally, as a clinician, one of the ultimate challenges we have is really, again, to undertake approaches that allow us to develop and test biomarkers using very small clinical specimens, and most commonly formalin-fixed paraffin-embedded tissues. And this is, again, an area where we've had multiple advances over the last few years and increasing capabilities, but we have to recognize the need potentially for the ability to look at DNA, RNA, and or protein on these small specimens. And so what I'd like to spend the next sort of 15 minutes or so is sort of talking about some of the research that we've done in melanoma and how these questions and challenges are being addressed. So as I mentioned, melanoma really has a very high level of oncogenic mutations, and what we've been able to identify are some of the key oncogenic pathways that are activated in this disease. And so this figure shows on the left the RAS-RAF-MEP kinase signaling pathway, on the right the PI3 kinase AKT pathway. So starting with the MAP kinase signaling pathway, what we actually know is that this is almost a universal event in melanoma. So approximately 50% of cutaneous melanomas have point mutations in the BRAF serine threonine kinase that increase its activity up to 200-fold. Another 20% of patients have activating mutations in NRAS, and these are mutually exclusive with those BRAF V600 mutations. And another 15% of patients have loss of function mutations in NF1. And so really between these and a few other genetic events, we know that activation of the RAS-RAF-MEP kinase signaling pathway is essentially a universal event that occurs in cutaneous melanoma. Now, early studies have shown in melanoma and other cancers that NRAS mutations, in addition to activating the MAP kinase pathway, can activate other pathways, and so this was actually the initial evidence for a role of the PI3 kinase AKT pathway in this disease. And what we've seen over time are rare activating mutations in the PI3 kinase and AKT kinases, and more frequently, complete loss of expression of the PTEN tumor suppressor gene, the major negative regulator of this pathway. And so overall, what we've identified is that a significant subset of cutaneous melanomas have concurrent activation of the PI3 kinase AKT mTOR pathway, as these events tend to particularly happen in tumors that have those activating BRAF mutations. Now, we actually, as I mentioned, now have multiple targeted therapies that are actually approved for patients with a BRAF V600 mutation. And again, these therapies can be very effective. What's very interesting, though, is that we've really developed a much better understanding of the impact of the MAP kinase signaling pathway on the anti-tumor immune response. So some of this work has been led by Dr. Jennifer Wargo, who showed first in vitro that in tumors that have an activating BRAF mutation, that inhibition of the MAP kinase pathway resulted in increased antigen expression on the surface of melanoma cells, making them more recognizable to the immune system. Shortly thereafter, Patrick Hu from MD Anderson showed in an in vivo model that blocking the MAP kinase pathway in a BRAF mutant melanoma cell would improve trafficking of therapeutic T cells into tumors. And again, Dr. Wargo and also investigators in Australia have shown in biopsies of BRAF mutant patients collected before the start of treatment and after 1 to 2 weeks of treatment that in metastatic melanoma patients treated with BRAF inhibitors and BRAF-MET combination, that we see with targeted therapy an upregulation of melanocytic antigens like MART1, as shown in the very top panel, and perhaps just as critically, shown in the lower panel, a massive influx of CD8-positive T cells into tumors. And so this data and functional studies have now led to multiple clinical trials that are ongoing in our field to evaluate the safety and efficacy of combining approved immunotherapy with the approved MAP kinase pathway targeted therapies, with really exciting results seen in early phase clinical trials. Now, as I mentioned, another active pathway in this disease in a subset of patients is the PI3 kinase AKT pathway, which is commonly activated by loss of the tumor suppressor PTEN. And my lab and Dr. Patrick Hu's lab collaborated to evaluate the impact of activation of this pathway on the anti-tumor immune response. Our initial in vitro work showed that co-culturing of tumor cells and immune cells with that loss of PTEN in the tumor cells made them directly resistant to T-cell mediated killing, and further in vivo studies in that same model that they had tested the BRAF inhibitor with showed that when we had loss of PTEN in tumors, we identified decreased infiltration of T cells into the tumors in these preclinical models. This was actually really validated in patients by looking at a large cohort of stage 3 melanoma patients as well as the melanoma TCGA cohort, where we found that tumors that had loss of PTEN had less immune infiltrates, including both CD3 and CD4 positive T cells. Now, T cell infiltrates have been strongly linked to sensitivity to immune checkpoint inhibitors, and particularly anti-PD-1. And so we reviewed treatment outcomes of metastatic melanoma patients treated at our institution with anti-PD-1 antibodies. This waterfall plot shows the maximal change in tumor size. The bars that go up mean the tumors were growing. The bars going down means that the tumors got smaller. The bars that are blue are reflecting patients who had intact PTEN expression in their tumor, and the bars in red reflect the outcomes of patients who had complete loss of PTEN. And as you can see, we saw a striking association with loss of PTEN with inferior responsiveness to anti-PD-1. And this resistance to anti-PD-1 was recapitulated in a GEM preclinical model in which melanomas express both the BRAF V600 mutation and have complete loss of PTEN. And in this model, we saw very little efficacy with anti-PD-1, anti-CTLA-4, or an OX40 antibody. Similarly, a PI3 kinase beta selective inhibitor was able to inhibit the PI3 kinase pathway in the PTEN null tumor cells without actually interfering with the immune viability or function. As a single agent, it did not have much activity, but when we combined those two agents together, we saw a marked improvement in the T-cell infiltrate into the tumors and much better anti-tumor activity. And actually, based on this data, we now have a phase 1, phase 2 clinical trial ongoing at MD Anderson, combining a PI3 kinase beta inhibitor with the anti-PD-1 antibody pembrolizumab in PTEN null PD-1 refractory patients. Now, this slide actually shows the patterns of PTEN expression that we see in melanoma. And particularly in the top left, we see an image of a tumor that has complete loss of PTEN expression in the tumor cells. The only cells that are staining positively are endothelial cells, which act as really an internal positive control for our PTEN immunohistochemistry. And we really see sort of a range of expression from absent all the way through normal to increased, with roughly a prevalence of sort of 20 to 30% for each of these patterns. And almost all of our tumors really show uniform PTEN expression. However, as we've really looked at hundreds of stage 3 and stage 4 metastases, rarely we've seen tumors that actually have much more heterogeneous expression. And this is an example of a tumor that really has areas that are very distinct with complete loss of PTEN expression on the right side of this tumor, whereas on the left, that area of the tumor really expresses very high levels of the PTEN protein. And these rare tumors, to us, are valuable tools to really try to better understand the biology and the immunology associated with PTEN loss. So this is data that we presented at the SITC meeting in 2017, where we went into a handful of these tumors and first actually did cord needle biopsies of these heterogeneous tumors to isolate regions that were PTEN expressing or PTEN null. We isolated DNA and performed next generation sequencing and DNA methylation. We then also isolated RNA from this tissue for RNA expression profiling. And overall, this actually suggested that the mechanism of PTEN loss was most likely genetic as opposed to epigenetic, and we did see differences in gene expression patterns. However, as Joe mentioned, the digital spatial profiling platform seemed like a really interesting tool to apply to these heterogeneous tumors to be able to actually look broadly at protein signaling pathways and the anti-tumor immune response on FFPE slides without actually sort of going through those types of dissections that we did. And also, again, the multiplex capability to allow us to look at multiple different markers on 1 to 2 FFPE slides. And so, again, on the left is shown sort of the schemata for how the digital spatial profiling works for protein profiling, with, again, the key being these tagged antibodies with the UV-sensitive linkers. And so on the right, this actually shows an example of one of these tumors with heterogeneous PTEN expression. And so the panel on the left side is actually the PTEN immunohistochemistry showing the region of the tumor that had PTEN protein expression shown in brown and labeled as PTEN positive, and at the bottom the PTEN null area where again we see an absence of staining. And so as Joe mentioned, we selected multiple regions of interest, or ROIs, in the PTEN positive and PTEN null regions. Those in the PTEN positive are the red circles seen in the upper part of the slide, and the ROIs in the PTEN null region shown down at the bottom in blue. And so we used one slide to evaluate a panel of immune markers and another slide to evaluate a panel of signaling pathway markers. And so this is actually shown on the left-hand part of the slide then, just our first initial analysis of the first tumor that we ran on the digital spatial profiling platform. So again, we had 6 regions of interest from the PTEN positive areas of this tumor, and these are reflected in the bar graph of the left with the 6 bars. And then on the right, the ROIs from the PTEN null region of the tumor. And so as a first validation, we were very interested looking at PTEN expression. We actually found that an antibody recognizing phospho-PTEN was a particularly robust marker that correlated very well with immunohistochemistry. We then started looking at activation-specific molecules such as phospho-AKT, and consistent with our previous studies in frozen tissues, we saw that the regions that had complete loss of PTEN had elevated levels of phospho-AKT, although what is also interesting, and again a unique capability of the digital spatial profiling platform, is we can actually see there's heterogeneity within the PTEN null region. Again, in the region that expressed high levels of PTEN, very low levels of phospho-AKT. And again, then moving further down the pathway to the level of phosphorylation of 4-EBP1, again, consistently we do see high levels of phospho-4-EBP1 in the PTEN null regions, but now we do actually see some other areas of the tumor where PTEN is expressed, where we do see high levels of this phosphoprotein. So again, I think on a single tumor level, sort of a nice illustration of how we could actually, again, look separately at the PTEN null and positive regions. And so we actually did this analysis on 7 tumors that had heterogeneous PTEN expression. And so again, just confirming the robustness of looking at PTEN expression, we saw again a marked difference in the PTEN expression as analyzed by digital spatial profiling, and we identified several proteins that were significantly upregulated in the PTEN null region, including phospho-AKT and phospho-P76K as expected, and we can also see sort of a series of signaling proteins that were downregulated or expressed at lower levels in the PTEN null region. And again, as Joe explained, we could also look at immune markers, and so again, similar to what we had seen in our immunohistochemistry studies, we saw downregulation of CD3, CD45, and CD8 in the PTEN null regions of the tumor, but also other interesting markers we hadn't looked at before, such as VISTA. And so again, I think looking at sort of individual heterogeneous tumors where we, again, looking at different molecular phenotypes, sort of a nice sort of proof of principle study of sort of the capabilities of the digital spatial profiling approach. Now, another area that we've been very interested in terms of molecular heterogeneity are brain metastases. So brain metastases are a huge clinical problem in many diseases, but they're a particular problem in melanoma. And indeed, among all solid cancers, melanoma actually has the highest risk of CNS metastasis, with up to 70% of patients showing evidence of CNS involvement on autopsy studies. Historically, these patients have had very poor outcomes with a median overall survival of approximately 4 months from diagnosis, and those outcomes are improving with our contemporary targeted therapies and immune therapies. However, we're consistently seeing that those treatments do not work as well in patients with brain metastases and in the brain metastases themselves as we see in extracranial tumors. And indeed, the brain is a common initial site of relapse for both targeted and immune therapies. And while historically much of the challenge of brain metastases has sort of been attributed to the difficulty of penetrating the blood-brain barrier, there really is very strong evidence now that brain metastases significantly disrupt the blood-brain barrier and that there's significant leakage of drugs into that space. Indeed, the BRAF inhibitor dabrafenib, which was not predicted to cross the blood-brain barrier based on preclinical studies, has actually very high response rates in melanoma patients with brain metastases. And what's really instead growing is an increasing appreciation, supported by data from multiple groups, showing that there can be unique molecular and immune features in brain metastases compared to non-CNS metastases. And we've done some work in this area. Our initial study back in 2009 was an experiment in which we looked at OCT-embedded or frozen melanoma metastases. We had looked largely at regional metastases, but we also did a pilot study of small cohorts of distant metastases resected from the brain, the liver, and the lung. We used H&E-guided macrodissection to isolate tumor-enriched regions and then did protein isolation, which we then ran on the reverse phase protein array platform at MD Anderson. And as, again, an initial hypothesis, we were very interested in looking at the correlates of PI3 kinase pathway activation. And so this graph shows the data for 3 different activation-specific markers in the PI3 kinase pathway. The blue bars show the levels of phospho-AKT serine 473, the red bars phospho-AKT threonine 308, and the yellow bars phosphorylated GSK3 alpha beta. The green bars, again, show the levels of expression of the negative regulator of this pathway, PTEN, and the data shows the median value for multiple tumors isolated from each of these sites. And although this was, again, a pilot study, a relatively small number of tumors, we saw a striking upregulation of activation-specific markers in the PI3 kinase pathway in the brain metastases compared to liver and lung metastases, and also significantly lower expression of PTEN in the brain metastases. Now, these tumors were from different patients, and so we did a subsequent study, and actually we went back to our tumor bank and identified brain metastases that had been resected from patients with metastatic melanoma as part of their standard of care, and specifically for patients who had also had non-CNS metastases resected from other sites. And so again, we used OCT-embedded tissue, H&E-guided microdissection to isolate proteins to run on the protein arrays. We also looked at hotspot point mutations and showed that they were very consistent between the brain metastases and the extracranial metastases in individual patients. And at the RNA level, we also saw very high level of similarity. But actually at the protein level, the striking difference that we saw was that every single brain metastasis had a higher expression of activation-specific markers in the PI3 kinase pathway compared to the extracranial metastases in the same patients. And this actually has been validated independently by a group in Germany by immunohistochemistry studies, again strongly implicating the PI3 kinase pathway in brain metastases. Now again, those studies were conducted with OCT-embedded frozen tissues, and over time, again, we've seen a rapid expansion in the capabilities of multiple technologies to work with FFPE specimens, which really opens up large number of specimens for analysis. And so we've actually collected a cohort of 98 brain metastases from 84 patients. We've used these tissues to do isolation of DNA and RNA. The results of those analyses will actually be presented at the 2018 AACR annual meeting for those of you who are interested. In terms of proteins, we've done some standard IHC markers, but again, this we thought was an interesting opportunity to again use the NanoString digital spatial profiling platform to characterize signaling proteins and the immune response in these clinical specimens. And in particular, as Joe mentioned, the ability to use masking so that we could separately look at the expression of those markers in tumor cells and in immune cells was something that again was very attractive to us in these highly heterogeneous specimens. And so this slide actually shows our first pilot experiment. This is actually a brain metastasis that we analyzed from a patient who had a brain metastasis and an extracranial metastasis. We selected now 12 regions of interest in each tumor to assess spatial heterogeneity. The data was then that we obtained was normalized to the number of cells in each ROI, again using the digital spatial profiling to look at our panels of signaling pathway proteins. And what we again did is use the masking, and an example of this is shown down in the lower panel on the left, where what we can see is we used S100B to recognize the melanoma cells, which are shown in green. We then used CD45, which is shown in red, to recognize the immune cells, and then we could use DAPI to actually count the number of nuclei. And so our initial analyses, again, wanted to focus specifically on the status of signaling pathways in the brain metastases, and thus in the S100 positive cells. And so down here at the bottom, we see the results for the signaling proteins in this single patient, where we again were able to separately measure 12 different regions of interest in the brain metastasis and 12 different regions of interest in the extracranial metastasis, and measure multiple phosphospecific proteins. And as indicated by the arrows, what we saw for multiple markers in the PI3 kinase pathway was that the expression of these activation-specific markers was significantly higher in the brain met from this patient, which are shown as the pink circles, compared to the non-CNS metastasis, which are the green circles there. And so overall, this again sort of identified upregulation of multiple phosphospecific markers in the PI3 kinase AKT pathway in brain metastasis. And in some ways, again, this was a nice confirmation of what we had seen before in our RPPA-based study, but to me really sort of jumped out in the fact that we were able to accomplish this with a single FFPE slide, the fact that we were able to assess intratumoral heterogeneity, which we're still working on, and the fact that we, again, didn't have to destroy any tissues so that we could go on and do other studies on this same slide. And so shown here, Joe mentioned, I think quite briefly, but we now are sort of working with NanoString on some pilot studies to, again, do the digital spatial profiling, but now not of proteins, but actually of RNA molecules. And so, again, this is being accomplished on a single FFPE slide, where we're now doing in situ spatial analysis of just over 1,000 RNAs, where, again, we can use the masking approach to separately look at the expression in the tumor cells with the S100 mask, and separately and independently in the immune cells that we see in each of these tissue samples using the CD45 mask. And so this is, again, a very exciting extension of the technology that we think will be quite powerful for our studies and studies of other investigators moving forward. And so just to summarize the data, again, it's really a very exciting time to be working in oncology as we develop much more effective therapies and as we develop really powerful tools to really sort of develop biomarkers that can be used to guide the therapy for our patients. And so I hope, again, I've explained that the fact that there is really strong evidence that oncogenic pathways in tumor cells can impact the anti-tumor response. And in data that I didn't show you, there's actually very strong data that the immune response will impact the efficacy of targeted therapy. And so together, this data really strongly supports the rationale for integrated analysis of both signaling and immune pathways in our clinical specimens. I think hopefully you've also appreciated the growing understanding that we have of the complexity of and the heterogeneity of both molecular and immune markers that we can see in tissues, both between different tumors and even within individual tumors. And finally, what we find so exciting is the increasing capabilities to interrogate molecular and immune features on small FFPE clinical specimens. And again, as we've demonstrated, again, a tremendous amount of information that can be attained even for single FFPE slides. And I think this, again, is providing tremendous potential and opportunity for new insights, new biomarkers, and ultimately new personalized approaches for our patients. So with that, I'd like to thank many of the people who worked on this project, our collaborators at NanoString, and the support that we had for this study. Thank you.
Thank you, Dr. Michael Davis and Dr. Joseph Beecham, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your questions into the box that appears on your screen and click on the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is for Dr. Beecham. What do you see as the potential advantages of digital spatial profiling capabilities?
Yeah, thank you very much for the question. I think probably Mike and I actually sort of highlighted very, very similar things in terms of the intrinsic advantages. And I thought of one that we actually didn't mention, so I want to add that to the list. But clearly being able to have the high multiplexing allows Mike to be asked those kinds of questions where what are the signaling pathways linked to heterogeneous areas of a tumor? That's a question that simply could not be asked in a quantitative way before we were able to do the kind of high multiplex work that the digital spatial profiler provides. So I think having the high multiplex capability, the unique capability of being able to mask out regions of tumor and tumor microenvironment, I think both of us highlighted that. This idea that you can actually make these measurements and that you actually haven't utilized any samples, so you get this reusability component associated with the technology is a huge advantage to be able to go and reinterrogate a sample. So we're really entering this is really a new type of capability inside the microscope. We're sort of bringing the power of molecular profiling into the spatial regime. And that's really the major advance we have here. The one that we didn't mention so much, and I'm just going to highlight here briefly, is, microscopy is sort of a specialized art. You got people that know how to run microscopes and don't. And I said highlight this digital spatial profiler. You don't actually have to be a microscopist at all. All you really do is you load the slides into the front of the digital spatial profiler. You close the door, and it's off. It runs automatically. It's all remotely controlled. You could have a different, it could be being controlled by a pathologist that's not even in your building. As a collaborator, it could be, halfway around the world and actually be helping you control the microscope or control the automated, the regions of interest. But the key thing is you don't have to be a microscopy expert to do this. If you're a molecular profiler and all of a sudden you really want to become, essentially doing state-of-the-art high-plex microscopy, you don't have to learn how to become a microscopist. It's all this instrumentation is basically automated. So anyway, I think that captures the strategic features that both Mike and I were trying to highlight in this talk. Thank you.
Yeah, and I think I'll just add to it. I mean, again, as a biologist, when we say that tumors have increased expression of a particular target versus another based on homogenates, we do always wonder, is this about sort of a specific subpopulation of cells that have very high expression whereas others don't, or is this something global? And again, really appreciate with this technology the fact that you can address this spatially, and actually not just for one marker, but as Joe mentioned, really for whole panels of markers at a time. The other thing that this actually allows us to do is actually look at the interactions between different markers, between different signaling pathways, that I think is really going to provide us with much better granularity about our understanding about the molecular biology, about the immunology, and about the heterogeneity of cancer.
Thank you. Our next question is for Dr. Davis. You talk about the challenges surrounding maximizing analysis of small FFPE sections. How does a platform like DSP impact research strategies and your everyday practices in the lab?
Yeah, it's a great question, and again, I think one of the most interesting aspects of the DSP platform. It's one of the things that in the studies we presented, we really were sort of highlighting, again, looking at heterogeneity of tumor cells, again, first in regions with intratumoral heterogeneity, and then with the brain metastases in tumors where we had high intermixing of different cell populations. One of the practical applications of this is actually using samples or biopsies where we actually have a lot of necrosis, and being able to actually, again, not let those parts of the tissue sort of contaminate your analysis. To be able to go in and spatially pick out sort of regions of viable tissue is another, I think, very practical advantage of this approach. I think overall, again, what we're really encouraged by is we, as in the field of melanoma, have found DNA-based predictors of outcomes, RNA-based predictors of outcomes, and protein-based predictors of outcomes. And as I was illustrating at the end, the fact that we're, potentially looking at a platform where you may be able to get DNA, RNA, and protein-based data really all at one time is really quite intriguing. And I think, in addition to really sort of having potential applications in research, I think ultimately that could have real implications for, clinical and diagnostic testing and hopefully for guiding patient care.
Thank you for that answer. Our next question is for Dr. Davis. How can the DSP platform help to develop tools that can be used to select patients that will benefit from immunotherapy?
Yeah, so this is obviously a very pressing question, not only in melanoma, but in multiple fields. And again, this is an area where we've seen the development of both DNA and RNA-based biomarkers, as well as some immunohistochemical stains. And again, I think that there's going to be an opportunity to be able to assess these things at the same time, or to be able to access multiple different parameters, again, using a very small amount of FFPE tissue. I think the other part that what we're going to see the ability to do is, again, many types of approaches, particularly RNA-based approaches, have been homogeneous, which really, again, end up being significantly influenced by what your relative proportion of tumor cells and immune cells are. I think where there's a real opportunity here is to be able to separately look to say, are there, again, markers in the tumor cells that tell us about will patients respond or not? Or separately, are there markers in those immune cells? And again, one of the things that we're particularly excited about is the ability to look at the status of signaling pathways not only in tumor cells, but at their status in the immune cells as predictors of outcomes. And our hope is that, again, doing those types of analyses will not only identify biomarkers that can predict which patients will or won't respond, but hopefully will also provide biological insights that explain why resistance happens, and again, provides rational strategies to overcome them.
Thank you for that answer. It looks like we have time for one more question. Our last question is for Dr. Davies. There is a growing number of RNA-based diagnostic tests. What are the advantages and disadvantages of doing testing with spatial resolution?
So great. I think that's, again, sort of a very important question. Again, I think that one of the things that's a real advantage of what we've sort of described here so far is the ability to do this with a single FFPE slide, the ability to know what it is you're profiling and to do that quite carefully. I think that, in terms of disadvantages, the part that we're going to be moving forward with is needing to validate those types of, that we're going to need to validate this moving forward as a new way to look, at least when we talk about things like RNA signatures. Certainly, again, I think in terms of looking at protein markers like CD8 positive cell infiltrates or PD-L1 expression, those are going to convert very easily. But I think that, again, this is a very powerful approach that we're going to be able to use moving forward.
I'd like to make one more comment there. So I'm sitting here looking at the last slide here, and I wanted to call out thanks to one other person that actually I see on your acknowledgments there, Mike, if you don't mind. Both Mike and I work very closely with a professor down there named Gordon Mills, and I just want to highlight and thank Gordon. Actually, Gordon and I are sort of co-inventors of this particular microscope, and I just want to sort of highlight and thank Gordon here publicly, so that it's been a really fun project, and working with Mike, it's been a close relationship between all the protein detection thing, and the whole team at MD Anderson is just exceptional, and it's been fun co-developing this technology together. Sort of technology, I got commercial technology development company with the best medical center in the world, and I think together we've come up with a pretty nice tool that's going to help patient care in the long term.
That's great. And it's been a pleasure to again work on this, and again particularly to work with people who are so invested again in generating a project that will really help patients. And again, I think this is a win-win for everyone.
I would like to once again thank Dr. Michael Davies and Dr. Joseph Beecham for their presentation. I would also like to thank NanoString Technologies for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.